everyone, I'm finally getting a chance to make the video. So you need to have the document out that looks like this. I, however, am going to go onto a blank SmartBoard document so that I can draw, because you can't draw on Word documents. So just to recap, on Monday in class, which feels like it was like several days ago, but it was really just yesterday, we learned about how cells, or an example of how cells can adapt or change their gene expression based on internal signals. And the example that we used was P53, and P53 is activated in response to DNA damage, okay? And so the internal signal is the DNA damage. And then P53 as a transcription factor binds to the enhancer of the P21 gene, allowing its transcription. And then we went through how P21 inhibits cyclin and CDK. And then if you inhibit cyclin and CDK, you stop the cell cycle. If you're confused about those details, we are going to go over them a little bit. We're going to go over them a little bit tomorrow. Okay? But we're now looking at external signals. Okay? So how do signals from outside the cell, okay, allow the cell to change its gene expression? So that's what we're looking at now. So I would write that down. So somewhere here, I would write that we're looking at external signals, okay, and how external signals can cause a change in gene expression. All right. So this is actually a review, okay, and it's going to seem pretty like it's going to be a review. So we'll see. So here we have a cell. Here's my cell. All right. And the cell, of course, has a nucleus, which I'm making oblong for a reason. You'll see why. And here, in the spider, are these signal molecules that are from an outside source. I'll give an examples of places we've seen this after I go through this. Now the question is how do these signal molecules, okay, tell the cell to change its gene expression? Well, you should remember that cells have receptors, okay? And if the receptor matches the ligand or the signal molecule, the ligand will bind. This will then cause a conformational change in the receptor, which usually activates it, okay? Then if you remember, this activated receptor will activate another protein. Okay, and now, remember this part up here was called reception. Now this protein will usually activate another protein, which will activate another protein. And if you remember, often these proteins are kinases because they activate each other by adding phosphate groups. Okay, this part of this process was called transduction, and if you remember, these are signal transduction pathways. Now, if you remember, usually the last protein that was activated, we said, and I was like, just believe it for now, is a transcription factor. Oh, really? We now know what these transcription factors actually do. So these transcription factors go into the nucleus, and I just drew this while the video was paused, because I hate drawing things while making the video. I make mistakes sometimes. And the transcription factor goes into the nucleus, and it binds to the region called the enhancer region. And remember, this transcription factor could also be called, in this case, a activator. And if you remember what happens when the transcription factor binds to the enhancer, is that bends the DNA, okay, and that allows RNA poly to more strongly attach to the promoter, and then it can transcribe the gene. Now, one thing I want you to realize is that this is an oversimplification of this picture, because there's not just one gene in this nucleus, there's like 30,000, but I'm zooming in on one. Okay? So, from this, okay, we were able to take an external signal, okay, these signal molecules, and through a signal transduction pathway and the activation of a transcription factor, turn on a gene, or change gene expression, or make a protein that is needed. Okay, now what are examples that we saw of this? The most common example that we saw of this was back when we were doing mitosis, and that these things were growth factors, if you remember. And they bound to here, and then one of these was called RAS, the other one was called RAF, and eventually a transcription factor was here, and it went into the nucleus, and if you remember, the growth factor, okay, caused the production of cyclin, okay, and if you remember from yesterday, cyclin binds to CDK and tells the cell that it's ready to divide, or it activates the proteins, the DNA replication proteins, so the cell can enter S phase and replicate its DNA to do mitosis. But the point is, without those details, is that outside external signal molecules work through signal transduction pathways by activating a transcription factor to bind to enhancer regions and turn on or off genes. Okay? Usually, in the case of growth factor, a lot of times growth factor is secreted by injured cells that want the cells to divide around it to replace the cells that have been injured. Okay. Now, the rest of the packet that follows, if you take a look, I'm going to talk you through it a little bit. So I got to find it. I have a million documents open. Where is it? Right here. Okay, so is an example of this, another example. Okay, and this example is looking at a hormone or a signal molecule in plants called ethylene. Okay, and ethylene causes fruits to ripen. Okay, so this example explains how ethylene causes fruits to ripen. Okay, so you need to read this paragraph, answer this question, and then if you take a look at this text describes these signal transduction pathways, and certain phrases are numbered. You need to, on this diagram, number where the phrases are occurring, and then there are three more questions to answer. This is going to be difficult. All right, I want you to do the best you can. All right, I'm now going to go make the rubric for your quizzes, and then grade your quizzes. It's going to be a long evening. Have a good night.